When we think of movement, animals often come to mind, but plants also exhibit fascinating movements. Unlike animals, which move their entire bodies, plants show movement through bending, twisting, and elongation of specific parts. These changes are responses to stimuli, factors in their environment that trigger a reaction. In this video, we'll explore how plants sense and respond to their surroundings, focusing on the unique ways they grow and adapt. We'll also compare plant and animal responses, setting the stage for a deeper dive into the world of plant hormones and their roles in growth and movement. Plants, like all living organisms, are constantly exposed to changes in their environment. These changes, known as stimuli, can be external, such as light, gravity, or water, or internal, like hormone levels. The way plants react to these stimuli is called a response. Unlike animals, which can move quickly to escape danger or find food, plants respond more slowly, often by changing their growth patterns. This ability to sense and react is crucial for their survival, allowing them to adapt to varying conditions and maximize their chances of thriving. One of the most significant differences between plants and animals is how they obtain food and respond to their environment. Plants produce their own food through photosynthesis, so they don't need to move to find nourishment. Animals, on the other hand, rely on plants or other animals for food, making movement essential. Additionally, animals have complex nervous systems and sense organs that allow for rapid responses to environmental changes. Plants lack these systems, resulting in slower, growth-based responses rather than quick movements. To adapt and grow, plants rely on chemical messengers called hormones. These hormones, also known as phytohormones, are produced in specific regions of the plant and play a vital role in regulating growth, development, and responses to stimuli. The main areas where growth occurs are called meristems, found just behind the tips of shoots and roots. Plant hormones help these cells divide and elongate, shaping the plant's overall form and function. Let's explore the different types of plant hormones and their unique roles. The concept of hormones was first introduced in 1902 by William Bayliss and Ernest Starling, but it was later refined for plants with the term phytohormone. These chemical messengers are distinct from animal hormones and are essential for plant life. They regulate everything from growth and development to responses to environmental changes. Understanding the discovery and naming of these hormones helps us appreciate the scientific journey that led to our current knowledge of plant biology. Auxins were the first plant hormones discovered, with the term coined by F.W. Wen in 1928. Derived from the Greek word meaning to grow, auxins are powerful growth stimulants effective at very low concentrations. They are found throughout higher and lower plants, especially in actively growing regions like shoot and root tips. The main natural auxin in plants is indole-3 acetic acid, IAA. Auxins play a crucial role in regulating various growth processes, making them fundamental to plant development. Auxins are responsible for a wide range of growth-related activities in plants. They promote the elongation of stems, roots, and fruits, delay the aging and falling of leaves, and encourage the growth of apical buds while slowing the growth of lateral buds. Auxins also induce rooting in plant cuttings, making them valuable for propagation. Remarkably, they can trigger fruit formation without fertilization, a process known as parthenocarpy, resulting in seedless fruits like certain apples, tomatoes, and bananas. Gibberellins are another important group of plant hormones. They exist in several forms such as GA1, GA2, and GA3, and are mainly found in meristematic regions like stem and root tips, buds, and seeds. Gibberellins are best known for promoting the elongation of internodes, breaking seed dormancy, and initiating germination. They also play a role in fruit growth and can induce parthenocarpy. In agriculture, gibberellins are used to enhance the size and shape of fruits like grapes and apples. Gibberellins have several key functions in plant development. They stimulate the elongation of stems, break seed dormancy to initiate germination, and promote fruit growth. Gibberellins can also delay the aging process in plants, keeping leaves and fruits fresh for longer. In horticulture and food industries, these hormones are used to improve the size and appearance of fruits, making them more appealing to consumers. Their ability to induce parthenocarpy is also valuable for producing seedless varieties. Discovered in the 1950s by Skoog and Miller, cytokinins are plant hormones that specifically promote cell division. They are produced mainly in root tips and transported throughout the plant via xylem cells. Cytokinins are abundant in germinating seeds, developing fruits, and embryos. Their primary role is to stimulate growth by encouraging cells to divide, but they also help expand cotyledons in seeds, break seed dormancy, promote chlorophyll synthesis, and delay leaf aging. Cytokinins are essential for various growth processes in plants. They stimulate cell division, leading to overall plant growth. In seeds, cytokinins help expand cotyledons, the first leaves that emerge during germination. They also break seed dormancy, allowing seeds to sprout under favorable conditions. Additionally, cytokinins promote the synthesis of chlorophyll, keeping leaves green and healthy, and help prevent premature leaf aging. By stopping apical dominance, they encourage the growth of lateral buds. Ethylene stands out among plant hormones because it is a gas at room temperature. Produced in fruits and some other plant tissues, ethylene acts locally, with its site of synthesis and action often being the same. Discovered by R. Gain, ethylene is produced in higher plants and fungi, especially in meristematic tissues. It plays a major role in reducing stem elongation, accelerating aging, and is widely used in agriculture to control fruit ripening and other processes. Ethylene is crucial for several plant processes. It reduces stem elongation and speeds up the aging of plant parts. Ethylene is best known for its role in fruit ripening, making it invaluable in agriculture for controlling when fruits are ready for market. It also initiates seed germination in peanuts, promotes sprouting in potato tubers, encourages root growth and root hair formation, and induces flowering in plants like mango. Its widespread use in agriculture highlights its importance as a plant growth regulator. Abscisic acid, or ABA, is known as a growth-retarding hormone. It is found in a wide range of plants including angiosperms, gymnosperms, pteridophytes, and some mosses. ABA is mainly located in the chloroplasts of leaves, with the highest concentrations in fruits and seeds. Its primary function is to inhibit plant growth and metabolism, helping plants survive stressful conditions. ABA also plays a key role in closing stomata and increasing the plant's tolerance to environmental stresses. Abscisic acid acts as a general inhibitor of plant growth, slowing down metabolism and preventing premature germination of seeds. It accelerates the aging and falling of leaves, buds, flowers, and fruits, a process known as abscission. ABA also stimulates the closure of stomata, the tiny openings on leaves, helping plants conserve water during drought or other stressful conditions. Because of its role in stress responses, ABA is often called the stress hormone of plants. Plants exhibit special movements called tropic movements in response to external stimuli. These movements are directional, meaning the plant part grows towards or away from the stimulus. The term tropic comes from the Greek word for turn, reflecting how plants adjust their growth direction. Tropic movements help plants optimize their exposure to light, gravity, water, and other factors, ensuring they grow in the most favorable conditions possible. Phototropism is the movement of plant parts in response to light. Shoots typically grow towards the light source, a phenomenon known as positive phototropism, while roots often grow away from light, showing negative phototropism. Auxins play a key role in this process by accumulating on the side of the shoot that is away from the light, causing those cells to elongate more and the shoot to bend towards the light. This adaptation helps plants maximize their ability to capture sunlight for photosynthesis. Geotropism, also known as gravitropism, is the growth response of plants to gravity. Roots exhibit positive geotropism by growing towards the Earth's gravitational pull, while shoots show negative geotropism by growing away from gravity. This ensures that roots anchor the plant and absorb water and nutrients from the soil, while shoots reach upwards for light. The balance between these movements is vital for the plant's overall structure and survival. Hydrotropism is the movement of plant parts in response to water or moisture. Roots are especially sensitive to water and will grow towards areas with higher moisture levels, a response known as positive hydrotropism. This adaptation ensures that roots are always close to available water in the soil, which is essential for the plant's survival. Shoots, on the other hand, typically show negative hydrotropism by growing away from moisture. Thigmotropism is the growth movement of plant parts in response to touch. Plants like sweet peas, cuscuta, and vines have tendrils that coil around objects when they come into contact with them. This one-sided contact triggers the tendrils to wrap around supports, helping the plant climb and access more sunlight. Thigmotropism is a remarkable example of how plants interact with their environment using physical contact. Chemotropism refers to the movement of plant organs in response to chemicals. A classic example is the growth of pollen tubes towards sugars and peptones secreted by the female gametophyte during fertilization. Fungi also exhibit chemotropism by growing towards areas with more food. This type of movement ensures that reproductive and growth processes are guided by chemical signals, optimizing the plant's chances of successful reproduction and survival. Let's see phototropism in action with a simple experiment. Take a healthy potted plant and place it in a dark room near an open window. After a few days, you'll notice the plant bending towards the window, demonstrating a positive response to light. This experiment visually shows how plants use phototropism to maximize their exposure to sunlight, which is essential for photosynthesis and growth. To observe geotropism, plant a few bean or gram seeds in moist soil inside a glass beaker. Over several days, the seeds will sprout and you'll see tiny seedlings emerge. The stems will grow upwards away from gravity while the roots will grow downwards towards gravity. This clear demonstration highlights the plant's ability to sense and respond to gravitational forces, ensuring proper orientation for growth. A clinostat is a device used in laboratories to study geotropism. By rotating a potted plant slowly, the clinostat ensures that all sides of the plant are equally exposed to gravity. When one plant is rotated and another is kept stationary, you'll notice that the stationary plant's shoot grows upwards and roots downwards, while the rotating plant shows no bending. This experiment demonstrates how gravity influences plant growth direction. To investigate hydrotropism, suspend moist sawdust on a wire netting and embed germinating bean seeds in it. As the seeds sprout, the roots initially grow downward due to gravity but soon turn upwards towards the moist sawdust, the only water source. This experiment shows that roots prioritize water over gravity, demonstrating positive hydrotropism, while shoots continue to grow upwards. In the hydrotropism experiment, roots initially follow gravity but soon change direction to seek out moisture. This behavior highlights the plant's ability to prioritize water over gravitational cues. For roots, water is a more effective stimulus than gravity, ensuring the plant's survival by keeping roots close to essential moisture sources in the soil. Throughout this video, we've explored the fascinating world of plant hormones and tropic movements. From auxins and gibberellins to cytokinins, ethylene, and abscisic acid, each hormone plays a unique role in plant growth and adaptation. Tropic movements like phototropism, geotropism, and hydrotropism allow plants to respond to their environment, ensuring survival and optimal growth. Understanding these processes helps us appreciate the complexity and intelligence of plant life. The knowledge of plant hormones and tropic movements has significant applications in agriculture and horticulture. By manipulating hormone levels, farmers can control fruit ripening, improve crop yields, and produce seedless fruits. Understanding tropic responses allows for better crop placement and irrigation strategies, ensuring that plants receive optimal light, water, and nutrients. These scientific insights contribute to more efficient and sustainable farming practices. As research continues, scientists are discovering new ways to harness plant hormones and tropic responses for improved crop production and environmental sustainability. Advances in biotechnology may lead to crops that are more resilient to stress, require less water, or grow faster. By deepening our understanding of plant biology, we can develop innovative solutions to global challenges like food security and climate change. Plants may seem stationary, but their ability to sense and respond to the environment is truly remarkable. Through hormones and tropic movements, they adapt, grow, and thrive in diverse conditions. By studying these processes, we not only gain insight into the natural world, but also find inspiration for innovation in agriculture and beyond. The world of plant hormones and tropisms is a testament to the complexity and resilience of life on Earth.